This podcast is supported by the Icahn School of Medicine at Mount Sinai, an international leader in research, education, and patient care. The medical and graduate school is part of the Mount Sinai Health System, one of the largest academic medical systems in New York City. Ranked among the top recipients of NIH funding, researchers at Mount Sinai have made breakthrough discoveries advancing the health of patients. Here, clinicians and scientists push the boundaries in cardiology, cancer, immunology, neuroscience, genomics, geriatrics, environmental medicine, and artificial intelligence. The Icahn School of Medicine at Mount Sinai, we find a way. This is the Science Podcast for January 8th, 2026. I'm Sarah Crespi. First this week, a quest to find Leonardo da Vinci's DNA. Contributing correspondent Richard Stone is here to talk about what researchers hope to learn from Leonardo's genes and the new field of arteomics. Next on the show, new evidence for poisoned arrows from 60,000 years ago complicates our picture of hunting during the Pleistocene. Researcher Sven Isaksson joins me to discuss the discovery of poisonous residues on microliths. These are tiny worked stone points used on arrows in an ancient rock shelter in South Africa. Arteomics is a growing field that brings together the latest science, from physics to microbiology to ancient DNA, with art history and preservation. The idea is to learn more about art from the past and the people that made it. This Week in Science, contributing correspondent Rich Stone wrote about a broad collaboration trying to bring these new tools to bear on the works of Leonardo da Vinci. Hi, Rich. Welcome back to the podcast.
Hi, Sarah. Good to be back.
Is that a pretty good definition of arteomics? Arteomics? What are you calling it?
I don't even know how to properly pronounce it. I've been calling it arteomics.
I looked it up. There's not even a Wikipedia page for it, so it is very cutting edge.
I'll tell you, that's Science Magazine for you.
Can you tell us more about this team, this Leonardo da Vinci DNA project, LDVP? What is their ultimate aim?
Well, their ultimate aim is to recover Leonardo's DNA. That's been the motivating force, the inciting question of the project since the very beginning when they got going in 2014.
And so the barrier here isn't we can't sequence DNA that's as old as he is, because we can go back much further. It's actually like a scavenger hunt. Like, he's left all these objects. They've passed through many people's hands.
It's complicated. Leonardo is a tough target in ancient DNA for a couple of reasons. One is it's not clear where his remains are. There is a tomb in France with some of his bones purportedly interred there, but his original grave had been disturbed and his bones had been mixed with others, and then they were reburied. So, in fact, it's not clear that the bones in the purported tomb of Leonardo are really his. It would take quite a bit of effort to get permission to sample those bones, to dig them up. Now, there is a lock of hair that may be Leonardo's that came from the same grave in the 19th century. Again, the authenticity is in dispute, but if those hairs do date from the time of Leonardo's death, then there could be an attempt to extract DNA from the hair. The other complicating factor, I should add, is Leonardo did not have any descendants.
There's this kind of circular thing like, oh, we found it on this art, we found it in this grave, we're not really sure that it's actually him. And how do you complete the circle and say, well, we have evidence that this is actually him? You can go to relatives if he had kids, but all we have is kind of cousins and then kids of cousins alive today.
Yeah, you're talking about a number of generations since Leonardo's day. He died a bit over 500 years ago. The potential sources of Leonardo's DNA are his artwork. He left behind fewer than 20 paintings definitively credited to him, but he had loads of sketches. He wrote voluminously, so there are manuscripts with his writing. These are all potential sources. There's been an amazing effort to document the Leonardo lineage. By that, I mean the Da Vinci's from his father down to the present day. Leonardo was born to a prominent notary and a servant girl. He was the only son, as far as people know, from his mother. Now, Leonardo's father was married to several other women, had a number of other children. So the genealogy has been traced to the present day, and there are several living descendants of Leonardo's father who have already provided samples that could be used to sequence their DNA.
This is a lot of generations, 20 generations.
We're talking more than 20 generations. Yeah.
What kind of markers can you trace over that long of a time when people are marrying and having kids with other people?
The most reliable way to trace the DNA back over generations and centuries in this fashion is the Y chromosome. So the male sex chromosome passed down from father to son. Researchers currently are focusing on the Y chromosome of the modern descendants and also from bones of Leonardo relatives, samples of DNA that have been extracted from Leonardo's sketches and from letters written by a cousin, and all try to converge on what's called a common Y chromosome Y haplogroup.
Let's go a little bit towards the art side here. This is super interesting. So, you're going to use DNA, say, oh, this is Leonardo's DNA. It's on this painting. So it's a little bit more tightly connected to him. So this provenance idea. But also, this is kind of improving our ability to analyze biological, quote unquote, contamination of art objects and say, hey, this has information for us.
The researchers have developed a pretty impressive platform for analyzing biological traces from artwork, sketches, drawings, etc. It's not just the human DNA, it's DNA from all the organisms that the object came into contact with over the centuries. So, for example, from a very famous Leonardo sketch called Holy Child, the research team was able to sequence a type of orange tree that was planted by prominent people in the Tuscany region of Italy during the period that the great master lived. This indicates they're on the right track. It kind of sets the environmental stage for all the other work done on these objects. There's also from a letter that Leonardo's cousin wrote, DNA was sequenced from that, and the malaria parasite turned up there. And malaria was a problem in that region in Italy during that period. You could really put together an intriguing environmental picture that gives you more confidence that the DNA you're pulling from these objects dates from that period.
Do we care about what is in Leonardo's DNA? So beyond just like sequencing it and saying it's an identity thing, do we care about how the DNA is configured?
Some people say that Leonardo was the smartest person in history, had the highest IQ. Genius is a very complicated thing. You're not going to get a single genius gene. But Leonardo was known for tremendous visual acuity. He could see patterns in moving water, like eddies and vortices, that today cameras can capture and you can slow down the speed. You can see these patterns after image processing. Leonardo could see with his naked eye. He could see the alternating beating wings of a dragonfly. Dragonflies have two sets of wings. When you see them flying around, like if you're a normal human with normal vision, it just looks like a blur. But Leonardo drew the two pairs of wings and noted that they beat in an alternating pattern. It's amazing he could pick up on that. One ultimate kind of goal of the project to recover Leonardo's DNA is to go beyond the Y chromosome to other chromosomes that could give clues to whether he had a rare allele that could have endowed him with this tremendous visual acuity.
And it could be at the level of the eye, or it could be at the level of the brain, right? We don't know because we do image processing. We have kind of a refresh rate on our vision. Any one of those things could be slightly different person to person.
Absolutely. It's all speculative, but maybe you could look in the descendants of the Da Vinci family to see if any rare alleles of, for example, potassium channels in the retina appear in contemporary generations.
You mentioned a few places that have been sampled from notebooks or letters or this one painting. Is the hope then to like go over the Mona Lisa and see what she has going on? Are there going to be more investigations of the paintings he's left behind?
The team certainly hopes that what they view as intriguing findings, the potential identification of the Da Vinci Y chromosome haplogroup, that is going to open doors for them, perhaps provide access to collections of letters and manuscripts that Da Vinci wrote that could contain samples of his DNA, because a lot of these samples of his writing have been kept under very strict conditions with limited to no access, the potential for modern contamination is much lower. So the team hopes to access these, gather more DNA, hopefully corroborate the initial findings that they have now, and hopefully make some progress on reconstructing other parts of the great master's genome.
There are a lot of techniques being brought to bear here. There's dozens of researchers from different disciplines, physicists, microbiologists, chemists, people who do ancient DNA. They're all kind of putting a hand in here. And that has resulted in techniques that they're very good at not disturbing the works or not destroying the works and still getting a sample that you need. And that can be applied more broadly. Da Vinci is, of course, a wonderful reason to kind of start this project. You know, as you say, like this giant in history, but it does spread out to other works, other artists, other parts of this field.
So it's worth noting that arteomics is not just DNA collection. It's a variety of biological and chemical techniques to extract useful information from historical objects. So there's been a big push at the Metropolitan Museum to expand this toolkit and apply it to mysteries that have long perplexed art curators. Just one example was a famous English painter named Thomas Gainsborough. He had claimed in some of his writings, he painted and he sketched to protect his sketches from the sulfuric London smog. He would dip his sketches in skim milk. And there was a really ingenious project that found milk proteins in Gainsborough sketches. This is just one of the applications of these new toolkits to try to solve longstanding mysteries.
We've kind of flicked at the fact that there's many different collaborators here, but there are physicists involved in this. What role have they been playing in kind of looking into Da Vinci, his DNA, or this bigger project?
You can talk about Leonardo's tremendous visual acuity, but how can you study it if Leonardo has been dead 500 years? One way to do it is to try to reconstruct the scenes that he sketched. For example, there are some very well-known Leonardo sketches on water flow, the formation of eddies, vortices downstream from the pier of a bridge, just tremendously detailed sketches he made. Two members of the Da Vinci project are actually hydraulic engineers, and they set out to reconstruct the flows that Leonardo was sketching as best as they could, and then try to estimate the frames per second that Leonardo's eye, would have had to have been able to process to be able to see these patterns. And they came to the conclusion that Leonardo would have been able to resolve frames per second at a much higher rate than people normally could distinguish.
He's seeing like at 100 hertz, but the rest of us are seeing like 30 to 60 hertz.
That's what the researchers proposed. It's, of course, very tentative, but based on their reconstructions, if there's a bell curve of what's called flicker fusion frequency, this ability to determine motion in frames per second. So he would have been at the far end of the bell curve.
I think normal television footage, movie footage is at 30 frames per second, thereabouts. You don't see each individual frame.
And I would emphasize it's not superhuman, but like I say, it's a bell curve. And there are potentially variants of potassium channels in the retina and other potential genes that allow people to differentiate frames per second at this extraordinary level.
That's so cool. And we both got to use our movie knowledge.
Yeah, this is not the usual fare for a science article.
Thank you so much, Rich.
My pleasure, Sarah. Thank you.
Rich Stone is a contributing correspondent for Science. You can find a link to the story we discussed at science.org/podcast. Stay tuned to learn about the earliest poison arrows found so far.
No one goes to Hank's for spreadsheets. They go for a darn good pizza. Lately, though, the shop's been quiet, so Hank decides to bring back the $1 slice. He asks Copilot in Microsoft Excel to look at his sales and costs and help him see if he can afford it. Copilot shows Hank where the money's going and which little extras make the dollar slice work. Now Hank has a line out the door. Hank makes the pizza, Copilot handles the spreadsheets. Learn more at m365copilot.com/work.
You said this place was steps from the water.
We just haven't found the steps yet.
How much did we save?
Enough.
Enough to get lost?
Or you could book a stay with Hilton.
Welcome to your oceanfront room, just steps from the water.
The Hilton sale is on now. Book on Hilton.com or the Hilton app and save up to 20% to get the stay you expected. When you want savings, not surprises, it matters where you stay. Hilton, for the stay.
When did humans first start using poisoned arrows to hunt? This week in Science Advances, Sven Isaksson and colleagues provided new evidence of this advance in hunting technology from as far back as 60,000 years ago in the Pleistocene. Hi, Sven. Welcome to the Science Podcast.
Hi, thank you. Great to be here.
This is such an interesting collaboration. When I looked at who was on this paper, it really is pulling from all different aspects of science. So what was your role in this work?
My role is as the organic residue analysis expert. So I came in sort of third in this work, providing the skills and knowledges to do these analysis for my colleagues.
And then they contributed their knowledge of hunting technology and psychology from tens of thousands of years ago and dating. So there's really just a really interesting mix of work coming together at this one site in South Africa. Can you tell us a little bit about the site, and then we'll get into the poisons?
It's called the Umlatusana Rock Shelter. It's in KwaZulu-Natal in South Africa. It's been excavated years ago in the late 1980s, I think. It's a rock shelter where the different soil layers are stratified. They're stacked on top of each other. The analysis that our South African colleagues has done on these layers show that it's a very intact stratigraphy. So there's this anthropological artifacts in stack of layers that are natural. What we've also done is to use dating method. It's called optically stimulated luminescence. That is kind of dating when the last time these sand grains saw daylight. And the posits where the microliths that we've been analyzing for this paper, where they were found, that layer was dated to approximately 60,000 years old.
And then we're going to go in a little deeper here and say there was poison on these microliths, which are used for hunting. So we'll get into how you detected that poison was present on these arrows.
Yes, so that's sort of a known phenomenon from historical times and also from earlier prehistoric finds that in southern Africa, poison has been used in hunting. It's been sort of the question of can we trace how far back this practice goes? Since this site was so well preserved, and one of our co-authors, Marlies Lombard, who has been going through the lithic materials and analyzing how far back you can find traces of bow hunting, she identified these points as projectile points or weapon points. And on some of them, she saw that there are actually macroscopic visible residues on them. Some have been analyzed to that they sort of contain adhesives, so they can be used to sort of glue the tips to the shaft. So what we've done now is that we collected microscopic samples, well, still visible, but very small samples from these residues, visible residues on the surface of 10 of those. So we selected the ones that looked promising for this sort of first study. And then we applied fairly standard procedures.
So you did gas chromatography.
Gas chromatography, mass spectrometry.
Right. GCMS, if you've ever heard of it.
GCMS, yes.
And you're not getting, this is poison. You're getting, this has poison that was sitting here for 60,000 years, degraded into something, and you could say that's a signature of the poison.
Yes. And actually, these components that we have identified, the two most significant of them, they are actually the active components from this poison. And what we don't see in any research paper, and especially not in this, is sort of the background work. We have been analyzing extensive body of ethnohistorical materials, and that was very crucial for this, involving sifting through hundreds of components to identify those with the chemical properties most likely to survive in very old contexts. And these crinane type compounds that we actually identified are among those.
How long has somebody been hanging around that you've been able to confirm before?
The oldest samples that we've analyzed in our group, it's like 1,000 years old. And already there you see a difference in how the trays look compared to the ethnohistorical materials. In other analysis, the earliest ones are like 7,000 years old or something like that. And that is, of course, something that I was concerned that it's such a big jump in time.
Yeah, absolutely.
It's a factor of 10. I was very sort of not having high hopes when we started to see if that we actually get something out of this. So I was positively not surprised, but I mean,
yeah,
you always have the hope.
How many of the tips that you tested had remnants on them of the poison?
It wasn't a subset. As I said, we collected samples that looked promising. We selected 10 samples, and on those, we found traces on 5. So I think that is, a very good result.
Definitely. And we don't know if that means that they only poisoned half of them or if that's just what survived.
No. And I mean, we selected samples that looked promising, and on those, we found traces on half of them. So there are some hundreds of points only at Umlatusana rock shelter. So there's more work to be done.
And you can say this is not the adhesive. This is a poison from a plant that we know about today that people have been using for millennia.
These two components, buffanidrine and epibuffanicine, they are known in the Amaryllis family of plants, but there's specifically one of them, the Bophone disticcia, that is commonly used still as an arrow poison for hunting. And it has a distribution that also, this site is sort of in the middle of it. So, and it's been around for a long time.
What are some of the other very old ones? What do we know about poison arrow technology kind of broadly?
We don't know very much. I mean, there's been analysis on materials from South America and from Southern Africa, but you need to have samples and you need to have technology that you have a material where you can get this signal from. So you have context, like in this case, stone points, weapon points, they're made from stone, because it's quite common all around the world to use with bone points, wooden points, and those materials don't preserve. The context is very important. So this might not be the oldest in the world, but it's the oldest we've found so far as we know.
It's the most findable of the oldest. Yes. Can we talk a little about what a hunt would have been like? How big is a microlith?
The size of these microliths is like the nail of your little finger. We think these are most probably arrows, and it's small arrows and small bows. It's not like these very strong ones. These points have been probably used for the hunting of quite small prey, like small antelopes, small bush pigs.
Very small. So they're not going to stab an animal and damage it very much unless they have a poison on it.
At least in historical times and in the ethnological materials that we have, and also in the prehistoric materials where we actually have found them together with shafts. They're made so that they should break off and stay in the wound in the animal and sort of distribute the poison. And the poison as such is not so strong that the animal would drop dead on the spot. They need to be understood in the context of persistence hunting, that you wound the animals and then track it until it lies down and you can take care of it. That's where my colleagues comes in with the sort of the psychology of it as well, since the effect of the poison is, I mean, it's biochemical rather than physical, so it's not necessarily visual or obvious. So this takes some cause and effect thinking to sort of figure this out. And also, what the poison does is that it shortens the time spent in tracking during persistence hunting. So it suggests capabilities of complex cognition and also like a developed working memory to be able to do this.
Well, you're also harvesting something and breaking it down to a useful material, applying it to something else that you built. It's a very complicated technology that we can now see going back so far in time.
Another thing that also is that this is evidence of the use of plants for other things than just food and tools. I mean, we know that people have been eating plants, foods, and we've been using plants for making tools. But I mean, here's the compound's efficiency as hunting poison depends on both preparation and dosage. In lower doses, it may very well have had medical applications. So we're into the sort of early evidence in the realm of using drugs, medicines, and poisons That's also fascinating. Yeah.
Could you look at other poisons that are used in other parts of the world, like as you said in South Africa or South America, and say, well, this poison looks like it would stand the test of time? Can you work backwards like that?
That's part of what we're doing now. I'm still going through this ethno-historical material that we have to look what kind of components can we expect to find. There's a lot of residues from different poisonous glycosides and compounds that are like water soluble that probably will not survive very long. And we've been looking at residues from that kind of poisons as well. And there might be survivors of those in these 1,000-year-old samples that we haven't analyzed before. But again, these are compounds compared to the ones we detected now that are much more susceptible to decomposition and leaching.
Thanks, Sven. This has been really fun to talk about.
Thank you.
Sven Isaksson is a professor of archaeological science at Stockholm University. You can find a link to the Science Advances article we discussed at science.org/podcast. And that concludes this edition of the Science Podcast. If you have any comments or suggestions, write to us at sciencepodcast@aaas.org. To find us on podcasting apps, search for Science Magazine or listen on our website, science.org/podcast. This show was edited by me, Sarah Crespi, and Kevin McLean. We had production help from Podigy. Our music is by Jeffrey Cook and Wen Koi Wen. On behalf of Science and its publisher, AAAS, thanks for joining us.
Meet the new iSIMS, the single talent acquisition platform that's fully future-ready. It works wherever and however you hire, with insights no one else offers. One solution that fits seamlessly into your existing tech stack, which is why IT and HR leaders trust it. With the best of enterprise-grade software and AI, iSIMS helps you identify and secure the right talent faster. iSIMS, powering exceptional hiring. Learn more at isims.com.